Am I audible and visible? Let me know. I think I am audible and visible. Am I audible? Hello everyone, welcome to my class. I am myself Dr. Deepthi Karya. I am the PhD Physiology and your educator for physiology. Okay, here today we'll discuss about neuromuscular junction. Topic neuromuscular junction. First of all, before starting the topic, I have few announcements. One announcement here, that is just a minute. Okay, fine. Here one announcement is, in this channel I'm going to upload videos for completing the whole syllabus. uh, Raj, I think here this is not NEET UG beta. I'm educating MBBS students, so you have to go to some NEET UG channel for the same. Okay, fine. So one announcement is, I am uploading videos of first year MBBS physiotherapy, nursing and homeopathy students. So if you are in any of this faculty, you are just watch these videos and I will going to complete the syllabus within 1 year. Okay, so please watch my videos daily. One thing. Second thing is, all my videos are free to access. Third thing is, if you wish to get all my notes, my all notes are available, and to get the notes you have to download the app Vesalius. Here this is the logo for the app Vesalius. This is available on the Play Store, and link for the same that is in the description box, so you can download the app and subscribe for the same. And if you wish to get hard copy, that is also available in the form of physiology notes. Fine. Now, so let us start with today's topic, neuromuscular junction. Okay, now, so starting with the structure, the name itself suggests neuromuscular junction. You can see here. Okay, neuromuscular junction, that is also known as myoneural junction, neuromuscular junction or myoneural junction. Here you can see skeletal muscle fibers, they are innervated by myelinated hallocation. They are innervated by myelinated motor nerve fibers. Okay, motor nerve fibers. This motor nerve fibers, they originate from motor neurons in the anterior horn cell of the spinal cord. You can see here, this is the motor neuron. Okay, and the junction between this terminal branch of the nerve fiber, that is motor neuron, and the muscle fiber, that is your neuromuscular junction or myoneural junction. Hello Kishan, welcome to my class. Okay, now action potential is transmitted, here you can see, from this nerve to the muscle. Right. Now, structure of neuromuscular junction, you can see at the neuromuscular junction there is no anatomical continuity, there is physiological continuation. Okay, physiologically these two parts are continuous, so this is physiological continuation, no anatomical continuity is there. Now next is now we'll discuss about the structure of neuromuscular junction. Sorry. So first of all, we have presynaptic terminal, this one. This is also known as axon terminal, this one, presynaptic. Terminal branch of the nerve fiber. Okay. Now, the cell bodies of this nerve fiber, they are located in the ventral horn. As you can see here, suppose this is your spinal cord. In the spinal cord, you can see here, this way. These are located, this is the cell body and this is the nerve fiber. This is your this nerve fiber, this one. Fine. Okay. So cell bodies are located in the anterior horn cell of the spinal cord, or it may be there in the brainstem. And this axons, which are myelinated, here you can see they reach to the skeletal muscle. When they approach to the muscle fiber, axon loses its myelin sheath. You can see here the myelin sheath is lost. Okay. And once it reaches to the muscle fiber. It loses the myelin sheath and it is divided into the terminals. Here, these are the terminals. Okay, axon terminals. You can see this are fine. Now, these terminals, they are known as terminal buttons or end feet. Okay. Now, these terminal buttons, what are these terminal buttons? These are expanded like a bulb. You can see here this one, like a bulb. They are known as terminal button and they are in close contact with, you can see here, sarcolemma of the muscle, and here that is through sarcolemma, that is cell membrane, it is in contact with the sarcoplasm. Okay. Now, in this synaptic or terminal button, you have this mitochondria, you have synaptic vesicles. These are synaptic vesicles, they contain neurotransmitter. Okay, this synaptic vesicles, in each this terminal button we have about 3 lakh synaptic vesicles. Okay, and neurotransmitter released here at the neuromuscular junction, that is acetylcholine. You can see here. Fine. Now, how this acetylcholine is synthesized? You can see that acetylcholine is synthesized in the mitochondria from, you can say here, acetyl coenzyme A plus choline. Okay, I will just show you one more presentation that is directing properly. I'll just give me 1 minute. I'll show you the other presentation that gives you. Sorry. Fine. This one. So this is your having this. Sorry. So some things are written here. So you can see here. In the mitochondria, this is the mitochondria. Okay. In the mitochondria, these are synthesized from acetyl coenzyme A and choline. Okay, acetyl coenzyme A plus choline. Okay, they form acetylcholine. Okay, and the enzyme required, I'm not going to write down, it is written here, that is acetylcholine acetyl transferase. Along with that, what we require is, we require glucose, we require ATP for the synthesis of acetylcholine from mitochondria. Okay. Now, after synthesis of acetylcholine, that is stored in the synaptic vesicles. You can see these are the synaptic vesicles in which this acetylcholine is stored. Okay, synthesized and stored. Okay, how many molecules are stored? Okay, these are stored in small packets, and these small packets are known as quanta. Okay, and each quanta that consists of 10 raised to 4 molecule. Okay, or you can say 10,000 molecules are there of acetylcholine. Fine. Now, this is pre-synaptic membrane, this one. Okay, and this is post-synaptic membrane. Okay, the pre-synaptic membrane, that is the membrane of the nerve. Okay, this one, that is terminal button, this one. And post-synaptic membrane, that is the membrane of the muscle fiber, this one. Fine. Okay, and here very important thing is 1:1. 1:1. One nerve fiber ends on the one motor end plate. Fine. One nerve fiber ends on one motor end plate. Now, between nerve fiber and the muscle fiber, we have a cleft. This one. This is synaptic cleft. Try to remember the normal width of the synaptic cleft, that is about 40 to 100 nanometer is the width of synaptic cleft. Fine. Very important thing here, in this synaptic cleft we have extracellular fluid. So all the constituents of extracellular fluid are there in the synaptic cleft, that is sodium ions, chloride ions, bicarbonate ions. They are in highest concentration in this synaptic cleft. Fine. Now, another is synaptic trough, or you can say this one, synaptic gutter. Here, these are axon terminal in the synaptic trough. This one, this part. Hello, Ashish. So, What is the synaptic trough or synaptic gutter? You can see here, this is your nerve, this is your muscle, fine. So this is the membrane of muscle, but how, why is it known as synaptic trough or gutter? Because below this end plate, this membrane of muscle that is thickened, and it gets invaginations are there, okay, that increases surface area, and this is known as synaptic trough. Very important thing here, we have receptors for binding with the acetylcholine, and the receptors are known as nicotinic receptor. Why nicotinic receptors? Because they are stimulated by acetylcholine and nicotine. Okay, another important thing here is this muscle fiber is covered here, this muscle fiber is covered by a layer of connective tissue here. This is basal lamina. Here in this muscle fiber we have basal lamina. Okay, this basal lamina, as it is written, that contains collagen, that contains glycoprotein and extracellular matrix protein. Okay, as well as very important thing here is the enzyme acetylcholine esterase. What is the role of acetylcholine esterase? Degradation of acetylcholine into choline and acetate. Fine. So this is about the synaptic trough or synaptic gutter. Now, next one, that is postsynaptic membrane or motor end plate or muscle membrane. This one. Okay, here you can see there is depressed membrane of the muscle fiber that forms a synaptic gutter or trough, as we have discussed. Okay, and it gives foldings. You can see these are the folds. Okay, with these folds are known as subneural folds. Another name, try to remember this name also, that is palisades. Palisades or subneural fold. Okay, what is the role of subneural folds in the membrane, muscle membrane? They increase the surface area, and they have nicotinic type of acetylcholine receptors, because they are, as I told you, stimulated by nicotine and acetylcholine and inhibited by curare. Curare is the competitive inhibitor of acetylcholine. Competitive inhibitor of acetylcholine means it competes with the acetylcholine to bind with the receptor. So if curare is present, acetylcholine cannot bind. Instead of that, curare binds. So that is competitive inhibitor. Fine. And another important thing, this nicotinic receptors which are present, they are ligand gated receptors. Ligand gated receptor means ligand is acetylcholine. When this ligand binds with this receptor, channel opens. The channel is for sodium ion. Okay, so sodium ions, they enter inside. Okay, and that results in transmission of membrane potential. Okay, now about 15 to 40 millions of acetylcholine receptors are present on this end plate. Okay, try to remember this value also. Now another is, this is acetylcholine receptor protein. This one, this is a protein having molecular weight, try to remember, 2,75,000. It is a gated channel, and whenever two molecules of acetylcholine binds here, this channel opens. Okay, and each channel consists of 2 alpha, beta, gamma and delta subunits. So 5 subunits are there. They have large diameter, pore diameter, and they lead to increased permeability of cations like calcium, sodium and potassium. Okay, when sodium enters inside, there is depolarization. Most of the time sodium enters. Why? Because in the synaptic cleft, as we have discussed, extracellular fluid is more, and this extracellular fluid has sodium concentration in large amount. Okay, so this receptors, when acetylcholine binds with this receptor, sodium channels open, sodium ions, they enter, and that results in depolarization. Fine. Now, what are the mechanism of neuromuscular transmission? There are, you can see, 6 steps. 1, 2, 3, 4, 5, 6. Which are this? Number 1 is, here action potential comes. First step. Second, this action potential causes release of acetylcholine. Second step. Third, acetylcholine binds with the receptor. Action. Fourth, initially miniature end plate potential means slight change in the end plate. And fourth, complete end plate potential is developed, and that results in production of action potential here in the muscle membrane. And lastly, acetylcholine gets degraded. Fine. Now, so first step we will discuss one by one. First step, that is arrival of action potential. You can see here, this is the action potential. As we all know, depolarization, repolarization. Okay, so this action potential comes at your presynaptic membrane. Fine. Hello Ashish. Now, as I told you, this presynaptic and postsynaptic membranes are not anatomically continued. Okay, anatomic, no continuation is there, only physiological. Physiological means for the transmission of action potential, there is continuation. Fine. Now you can see here, first of all, action potential comes here. Now, when action potential comes to the presynaptic membrane, calcium channels here, they get, you can see, here also, they get opened up. So calcium enters inside the presynaptic terminal. Fine. Now, when calcium enters from extracellular fluid into the presynaptic terminal, that causes exocytosis of acetylcholine from the vesicle. Fine. You can see here. So because of increase in the calcium ion in the cytoplasm here, okay, you can see this calcium ions, they cause your exocytosis of acetylcholine. Very important thing here you have to remember is there is interaction between synaptobrevin and syntaxin. What is synaptobrevin? Synaptobrevin, that is a protein present in the synaptic vesicle here. Okay, and syntaxin, that is a protein present in the neuronal membrane. So because of interaction between this synaptobrevin present in the vesicle and syntaxin here at the neuronal membrane, this vesicle gets fused with the neuronal membrane, and then it causes exocytosis of acetylcholine in the synaptic cleft here. Fine. Now, so you can see here, normally when one action potential comes, about 60 to 200 vesicles, they get ruptured, and one vesicle contains 4,000 to 10,000 acetylcholine molecules. Okay, so another thing, this is known as quanta. Why quanta? Means quantal release. Suppose one action potential comes, then this many vesicles and this many molecules of acetylcholines are released. Okay, and they enter in the synaptic cleft. Okay, and when two molecules of, as I told you, acetylcholine binds with this ligand gated channel, then it causes opening of the channel and entry of sodium ions. Fine. Now, so this is about your first step. That first step, as we have discussed, arrival of action potential, calcium entry, and bursting of the vesicle. Now there is a time for the action of acetylcholine. Now your acetylcholine is there in the synaptic cleft. Okay, and acetylcholine increases permeability for the sodium and calcium ion. Now you can say that negative ions cannot enter. Why? Why negative ions cannot enter here through this channel? Because already these channels are negatively charged. Acetylcholine receptors are negatively charged, so they repel negative charges. Okay, so because of entry of positive ions, initially there is end plate potential. What is end plate potential? That is local, as we have discussed yesterday. This is also known as graded potential. Graded potential, they are local, they will not pass non-propagated, okay, graded and summated potential. Okay, and they will not follow all or none law, so they are opposite to action potential. Okay, action potential have the property they are propagated. Okay, non-graded means they follow all or none law, and no summation is possible. Okay, but the here slightly change in the membrane potential, it will not be the action potential. This all are summated. Fine. So you can see, when you can see here, when small quanta of acetylcholine is getting released, around less than 10 raised to 6 molecule. Okay, there is slight change in the membrane potential. Sorry, you can say here, this membrane potential increases by only 0.5 millivolt. Very important thing, this is known as miniature end plate potential. Okay, when, very important thing here is, when small quantity of acetylcholine is released, then there is miniature, means minimum end plate potential is developed, and that is only change in the 0.5 millivolt magnitude of the potential. Fine. At rest also, this kind of 0.5 millivolt change in the membrane potential like this found. Why? Because of the movement of exoplasm here. Okay, exoplasm in the resting condition also Brownian movement of exoplasm is there. Okay, so because of that, slightly small amount of acetylcholine is released here, and when more and more quantity of acetylcholine is getting released, that produces miniature end plate potential, and this miniature end plate potential get added, and that produces end plate potential, and when this end plate potential getting added, that produces giant end plate potential, and that is converted into action potential. Okay, you can see here, this is end plate potential, and this one only this. I'll just show you. I'm erasing all the inks. You can see here, this is end plate potential, only this much. Once it reaches to threshold, that produces action potential. Fine. So generation of action potential. Okay, when the depolarization is 12 millivolt, up to 12 millivolt, but not reaches to the threshold, then there is only end plate potential. But when it reaches to the threshold, then that produces action potential. Fine. Now your resting membrane potential of the muscle is minus 90 millivolt. Okay, and threshold is minus 60 millivolt. Means when the membrane potential reaches to minus 60, then it generates action potential here. Fine. Now, then you can see this action potential has the property of propagation. Okay, it propagates along the muscle membrane and it causes contraction of the whole muscle. Okay, you can see here, this is the schematic diagram how action potential travels at the neuromuscular junction. Fine. So I'll show you. You can see here, once action potential is coming here, okay, calcium channels they open. Okay, calcium ions, they cause release of acetylcholine. Okay, this acetylcholine binds with the receptors present on the postsynaptic membrane here. Okay, you can see two molecules, they bind and they cause opening of the sodium channel and they produce action potential. This gets propagated on the membrane. Fine. Another is, after acetylcholine completes its action, you can see here, it gets removed. It gets broken down by the enzyme acetylcholine esterase. Okay, I'll show you that. I'll just show you this one. Acetylcholine, that is broken down by, within 1 millisecond, it is destroyed by acetylcholine esterase. Okay, this acetylcholine esterase is present in the matrix of synaptic cleft. Okay, and what is the importance of this acetylcholine esterase? If it is not present, what happens? Then continuous stimulation that results in continuous contraction of muscle. To prevent this, we have to remove this acetylcholine. Fine. Now, this acetylcholine is converted into choline and acetate. Now, this, what happens to this choline and acetate? Acetate diffuses to the extracellular fluid. It will go to the extracellular fluid, and choline is taken back by the presynaptic membrane here, and it is reused for the synthesis of acetylcholine. Fine. So acetylcholine converted into choline and acetate. Acetate will pass through the extracellular fluid and diffuse in the surrounding extracellular fluid, and choline is reused. Fine. So this is about the fat of the acetylcholine. So this is about today's topic. Now let us discuss few question answers, few MCQs. Okay, I'll just, let me start this one. Okay, I'll just give you a few question answers, MCQs. Okay, fine. So let us start. If you are watching me live, you can give me answer, and if you are not live, you are just watching recorded video, then also you can write down your answers. Okay, starting with the first one, width of the synaptic cleft. We have already discussed how much is the normal width of synaptic cleft. One, that is 10 nanometer, 20 nanometer, 50 to 100 nanometer, and more than 100 nanometer. We have discussed, if you watch the video, you can easily answer, that is 50 to 100 nanometer is the width of the synaptic cleft. Okay. Next question is, synaptic cleft is filled with a fluid which is rich in sodium, potassium, phosphate and protein anions. As I told you, in the synaptic cleft, this is presynaptic membrane, this is postsynaptic membrane, we have extracellular fluid. Okay, and extracellular fluid is rich in sodium ions. So this is your answer. Okay. Next is, each synaptic vesicle, this is your presynaptic membrane, postsynaptic membrane, okay, vesicles store how many acetylcholine molecules? 10 raised to 2, 10 raised to 3, 10 raised to 4, 10 raised to 5. Okay, that is, we have discussed 10,000 molecules or 10 raised to 4 molecules. Fine. Which enzyme is involved in the synthesis of acetylcholine? Choline acetyltransferase, acetylcholine esterase, pseudocholine esterase and glycine. We have discussed this acetylcholine esterase, that is for degradation. Okay, and choline acetyltransferase, that is for synthesis. So your enzyme for synthesis is choline acetyltransferase. Okay. Next is spontaneous release of acetylcholine at neuromuscular junction because of Brownian movement of exoplasm we discussed. What is produced? Miniature end plate potential, action potential, post-tetanic potential, resting membrane potential. Okay, and your answer is miniature. That is just change in 0.5 millivolt of the membrane potential. That is, it produces miniature end plate potential. Fine. Next is, which of the following is the effect of acetylcholine through nicotinic receptors? We have discussed nicotinic receptors are present at neuromuscular junction. Okay, so what are the actions? One, that is, it decreases heart rate, secretion of saliva, pupil constriction or contraction of skeletal muscle. It is obvious because neuromuscular junction, so it causes contraction of skeletal muscle. Fine. Which of the following statement is not correct regarding release of acetylcholine at neuromuscular junction? Increased sodium ion movement inside the muscle cell produces end plate potential, always cause muscle fiber to contract, and it is followed by rapid destruction of acetylcholine. Okay, here incorrect answer, that is, it will not always produce end plate potential. Okay, because sometimes if the small amount of acetylcholine is released, that change in the membrane potential only 0.5 millivolts, so it produces miniature end plate potential and not produce end plate potential. Fine. End plate potential is characterized by propagation, all or none law, depolarization, and none of the above. Propagation and all or none law, these two are the properties of action potential. Okay, so this is answer is depolarization is the property of end plate potential, graded potential. Fine. Now, amplitude of miniature end plate potential. As I told you, small quantity of acetylcholine is released. So how much resting membrane potential changes? Only, we have already discussed, 0.5 millivolt. Fine. And last one, nicotinic acetylcholine receptors. They are found on presynaptic membrane, postsynaptic cleft, motor end plate and postsynaptic membrane. We all know that here presynaptic membrane, here is the postsynaptic membrane, and the receptors are present here on your postsynaptic membrane. So this is your correct answer, postsynaptic membrane. Okay, so this is all about our MCQs from today's topic. Okay, thank you all. I repeat that if you wish to get a soft copy of all my topics, you can download the app Vesalius. Link for the application as well as link for my book, they are given in the description box. Thank you, Kishan. Okay, beta. So you can download and you can subscribe in this application as well as you can order the book also. Thank you so much. If you like my video, you can like it, you can share with all your friends, subscribe my channel Dr. Deepthi Thakkar Karya, and hit the bell icon please, because if you hit the bell icon you can come to know that madam is live, madam is online. Okay, class start हो गई है तभी पता चलेगा. So please subscribe and hit the bell icon. Thank you.